Hello everyone. Today we're going to have the second part of the blood biochemistry, and mainly today we'll focus on hemoglobin. Basically, we'll learn about the structures of hemoglobin, their properties, the clinical scenarios that can arise because of abnormal hemoglobin, certain diseases. We'll learn about them in details. So let's begin. Now, we have learned from the previous lecture that hemoglobin is the most abundant porphyrin containing compound. If you remember, porphyrin, uh, in the last chapter or the last class, we have learned about it. So basically, our hemoglobin is the most abundant porphyrin containing compound, and it's made up of 4 subunits, which give its a tetramer structure. 4 subunits together combine to form the structure of hemoglobin, and each of these subunits will have this heme group that we have discussed previously, and will also have a polypeptide change. So the structure of hemoglobin will look somewhat like this. If you can see here in this diagram, it will have 4 subunits, and in the center of these subunits, if you can see here, we have our heme group attached to the iron, and each of these subunits will also have the polypeptide change. All of these subunits will have it. Now, the polypeptide chains which we have in the hemoglobin are 5 kinds basically. We have alpha, beta, gamma, delta, and epsilon. In total, there can be 5 kinds of polypeptide chain. The alpha chain is made up of 141 amino acids. The rest that we have, beta, gamma, delta, and epsilon chains, are made up of 146 amino acids. So you can see here there is a big difference among the polypeptide chain. The alpha chain will have fewer amino acids compared to the other chain. The gene for this polypeptide chain of hemoglobin are also known as globulin genes, as you can see here. The genes that will code for this polypeptide chain. Now, these genes are found in clusters of 2 different chromosomes. The alpha cluster is present on chromosome 16, and the beta cluster is found on chromosome 11. Okay, so it's important you should know that in which chromosome number we'll find the gene that codes for this polypeptide. Okay, so keep in mind that the alpha cluster will be found in the chromosome number 16, whereas the beta cluster will be found in the chromosome number 11. We know that hemoglobin synthesis occurs in the red blood cells basically. As a precursor, the synthesis of the hemoglobin will take place in the red blood cells. It begins in pro-erythroblast stage of the red blood cells. And when the red blood cells reaches the erythroblast stage, 65% of the hemoglobin synthesis is already completed. The remaining portion will be completed at the reticulocyte stage of the RBCs. Now, part of the synthesis of the hemoglobin also will take place in the mitochondria. See here, the heme group, part of it will also take place in the mitochondria. Now, the synthesis will stop when the mitochondria will disappear from the red blood cells. We know that in a mature red blood cells, we don't have any mitochondria. But in pre-stages, that is erythroblastic stages and reticulocytic stages, okay, during that time mitochondria can be present. The globin protein part of the hemoglobin are synthesized on ribosome located in the cytoplasm. The whole process of synthesis of hemoglobin and globin are synchronized, so they will take place in a synchronized manner. The alpha 2 beta 2 hemoglobin chain that is found usually represents the adult hemoglobin, also denoted by HB capital A. In a normal adult, majority portion of the hemoglobin will be of this kind, will have 2 alpha chain and 2 beta chain. A minute portion of the hemoglobin will be in the form of HbA2, that is 2 alpha chain and 2 delta chain. Okay, but majority portion will be having the HB capital A. The alpha 2 epsilon 2 form of the hemoglobin is known as embryonic hemoglobin. The alpha 2 gamma 2 form is known as fetal hemoglobin, or also we call it HbF. In fetus, this is the major type of hemoglobin that you will find. But in some adults, you can also find fetal hemoglobin, but in a very minute amount. So, in summary, the whole structure of hemoglobin, if you consider, will be mainly having a secondary structure in the globule chain, that is alpha helix. If we have learned the structure of protein, you know that there are several stages: primary, secondary, tertiary, and quaternary. Okay, so the major secondary structure that we'll find in hemoglobin is our alpha helix structure. In beta, gamma, delta, and epsilon chains, there will be 8 alpha helical regions, and they will be named as from A to H. In alpha chain, however, you will find 7 helices instead of 8. The helix D will be missing in cases of the alpha chain. In cases of alpha chain, we will not have the D zone that we are talking about. Okay, in case of beta, gamma, delta, and epsilon, we'll have A, B, C, D, E, F, G, H, total 8 zones, okay, of alpha helices. However, for alpha chain of the hemoglobins, we'll only have 7. So the structure will look somewhat like this, as you can see here. A, B, C, D, E, F, G, H. In total, 8 helices will be found in a typical hemoglobin structure, which is of beta, gamma, delta, and epsilon. If it's alpha chain, we won't have this zone. The D zone will be missing in that cases. In the center of each hemoglobin or a hem group, we'll find a ferrous ion. The ferrous ion will have 6 electrons in its outermost part, and among those 6, 4 of them will link the iron to the 4 nitrogen of the heme group. So usually hemoglobin will exist in 2 forms basically. In a normal thermodynamic conformation, hemoglobin will exist in 2 forms. The 2 forms are known as T for tense and R for relax. When oxygen binds to the hemoglobin, we know that hemoglobin's main function is to carry oxygen, right? You all know this. So when the oxygen binds to the hemoglobin, it becomes relaxed. The hemoglobin changes its shape into a relaxed state from the tense state. So during this process, from converting from tense state to relaxed state, what will happen? You'll see a pair of alpha and beta subunit would rotate by 15 degrees. And the gap in between these 2 beta polypeptide chain will become narrower when oxygen will attaches to the iron. So there will be fewer gaps in between them. Now, this process of oxygen binding to the hemoglobin is regulated by an important regulator. We call this regulator 2,3-biphosphoglycerate. In short, we call it 2,3-BPG. And it's a very important regulator because it regulates the oxygenation and deoxygenation of hemoglobin. This 2,3-biphosphoglycerate will be formed in the erythrocyte at first as 1,3-biphosphoglycerate. And if you have studied the carbohydrate metabolism, you have seen that it's an intermediate of glycolysis, 1,3-biphosphoglycerate. So, and hemoglobin, as you can see here, having 4 subunits. For example, this one is having 2 alpha chain and 2 beta chain, but in the center you can see they have a hollow-like space. This is where we have the ferrous ion, and this is where the oxygen will ultimately bind. When binding takes place, the hemoglobin will change its shape by rotating the alpha and beta chain to 15 degrees, thereby will change from a tense state to a relaxed state. Now, when we have a very high concentration of 2,3-BPG or biphosphoglycerate, what will happen? This 2,3-biphosphoglycerate will come and bind to the central cavity. Now, what happens if 2,3-BPG binds to the central cavity? So when the 2,3-BPG will bind to the central cavity, it will do the reverse what oxygen do. It will change the hemoglobin from relaxed state to tense state. And as it happens, it will release the oxygen from hemoglobin. So at first what is happening? When oxygen binds to the hemoglobin in the central cavity, its conformation is changed from tense state to relaxed state. But if the body needs oxygen now, because oxygen, if my body will need oxygen, we'll need to extract it from the hemoglobin. So hemoglobin needs to release this oxygen, right? So in order to do that, what happens when we have a very high level of 2,3-biphosphoglycerate, it will bind to this central cavity, and thereby it will change the hemoglobin conformation from relaxed state to tense state. So there will be changes in shape. As a result, oxygen will be released. Now, these 2,3-BPG usually will bind to the beta chains by salt bonds or ionic bonds, and it will help in the release of oxygen from hemoglobin. How? By converting the hemoglobin to T form from R form. So when our body needs oxygen, there will be more conversion of 1,3-biphosphoglycerate into 2,3-biphosphoglycerate. This will allow the 2,3-BPG to bind to the central cavity and hence help us to release the oxygen. Just the opposite will take place when we have a very high amount of oxygen. If we have high amount of oxygen, okay, oxygen will bind to the central cavity of hemoglobin, change it from T to R form, and thereby will make the gap narrower between the beta chain. And if the gap becomes narrower, okay, there will be no space for our 2,3-biphosphoglycerate to bind. Okay, so we don't need to release oxygen because we already have a higher oxygen availability, so we would not need to release the oxygen anymore. Okay, so this is how basically the oxygenation and deoxygenation of the hemoglobin process are regulated. There is one more important thing we should know about this process of binding is that it takes place by the mechanism of something known as cooperative binding. So we know that there are 4 subunits of hemoglobin. Now each one of these subunits will bind to one oxygen molecule. So one hemoglobin molecule can bind to 4 oxygen molecules. Okay, so when the 4 oxygen molecule binds to the hemoglobin, it looks something like this, as you can see here, when it binding to the hem group. Now binding of one oxygen molecule will facilitate or encourage the binding of oxygen to the another oxygen molecule. So this facilitation, that is binding, once one oxygen atoms bind, it facilitates the binding of another, and as it happens, this is known as cooperative. That is, they will cooperate with one another, that is, encourage one another to bind. Okay, so you'll see that binding of the one oxygen molecules to hemoglobin will facilitate the binding of the another oxygen molecule, and this is known as cooperative binding. And this is the reason why we have a sigmoidal curve. When you check oxygen dissociation curve, if you have learned already in physiology, you'll see that oxygen dissociation curve is in a sigmoid shape, something more like an S. That is because of this cooperative binding of the oxygen atom. Okay, so something like this. The cooperative binding is not shown by myoglobin, however. We have learned from the previous class that myoglobin is a monomer. Oxygenated myoglobin will only release oxygen only when oxygen tension is very low. So the in oxygen dissociation curve, the myoglobin will not show the sigmoidal shape. It will look something like this, as you see here. However, hemoglobin will have more an S-like shape, sigmoidal shape. Okay, now our hemoglobin have several derivatives. That is, it can form several derivatives. It includes oxyhemoglobin, carboxyhemoglobin, methemoglobin, and sulfa hemoglobin. First one, oxyhemoglobin. is, you already know, is the oxygenated form of the globin. It's bright red in color and is transported to tissue in the form of oxyhemoglobin. Next one, carboxyhemoglobin, it will combine with carbon monoxide, and as it combines, it forms carboxyhemoglobin. The affinity of the hemoglobin for carbon is much higher than that for oxygen. So carbon C hemoglobin you'll see cherry red in color. You'll see if anybody is poisoned with carbon monoxide, if somebody is stuck in a airtight room and if there is carbon monoxide gas released, and the patients who are being poisoned with carbon monoxide, you'll also see that their skin, their face turning into cherry red color because of this. Because of why? Because carboxyhemoglobin is cherry red in color. Now, the structure of carboxyhemoglobin is much more stable compared to our oxyhemoglobin. Now, once the carboxyhemoglobin is formed, that is when carbon monoxide have bound to the hemoglobin once, it will become very difficult to separate them. And as carboxyhemoglobin is formed, this will also decrease our body's or blood's ability to carry oxygen. The next derivative is methemoglobin. There are certain drugs and chemicals that can oxidize the ferrous ion of hemoglobin to ferric ion. Drugs such as sulfonamides, such as nitrites, they can very easily cause the oxidation and ultimately convert the hemoglobin into methemoglobin. Now, methemoglobin will have a brownish red color. Some methemoglobin can be formed inside the body by our normal physiological process that takes place, but that is very minute amount. Our RBCs have enzymes called methemoglobin reductase and glutathione. So even if there is methemoglobin formed in the body by physiological processes, they will be reduced immediately into hemoglobin, and our RBC will be capable of doing so because the amount will be very minimal. Now, the methemoglobin, unlike our oxyhemoglobin and carboxyhemoglobin, they cannot bind to oxygen, but they can bind with something called cyanide. And if they bind with cyanide, they will form something called cyan-methemoglobin. Okay, so that's why if we have any patient who have been poisoned with cyanide, we can use methemoglobin or this property feature to treat those patients. As methemoglobin can bind with cyanide, okay, so it can be treated that way. Let's see how it is done. So if the patient comes with the signs and symptoms of cyanide poisoning, the patient at first is giving something known as sodium nitrite and sodium thiosulfate. Sodium nitrite has the ability, we have just seen, to convert hemoglobin into methemoglobin. Now, methemoglobin will bind with cyanide to form cyanomethemoglobin, which is less toxic, and the sodium thiosulfide that we are giving, this will react with cyanide to form something called sodium thiocyanate. In this way, we can reduce the toxic effect of the cyanide poisoning. The fourth derivative is called sulfur hemoglobin. Now, sulfur hemoglobin, okay, this is basically what this is also sulfonamides and H2S sulfur hemoglobin. Okay, hydrogen sulfide can convert the hemoglobin into sulfur hemoglobin. They will have a dirty brown color, and also just like the previous one, methemoglobin, they cannot bind with the oxygen, but they will persist in the red blood cells throughout the lifespan of the RBCs. So these are some derivatives. Now let's check out some abnormal hemoglobins. Now abnormal hemoglobins can be formed as a result of mutation of the gene. Which gene? The globulin gene. Most commonly, a single amino acid is affected and substituted in this mutation. There are hundreds of mutant hemoglobins which have been discovered. Most of these mutant hemoglobin can function normally or near normally. If they can function normally or near normally, we'll call these hemoglobin variant hemoglobin. Okay, we will not call them abnormal hemoglobin, but we'll call them hemoglobin variant. Why? Because they still can function either 100% or near 100%. That's why we're calling them variant hemoglobin. Okay. Now, in those cases where the mutation have resulted in impairment of the function of hemoglobin, maybe because the mutation have taken place in an important amino acids, and if the hemoglobin have lost its functioning ability, then only we'll call it abnormal hemoglobin. You see, in some mutation, it can take place in critical region, so this may impair the hemoglobin function. And if such happens, we're calling them abnormal hemoglobin. And if any disease develops because of these abnormal hemoglobins, we'll call them hemoglobinopathies. That is, diseases of the hemoglobin. Okay, some of the examples of hemoglobinopathies includes hemoglobin S, hemoglobin M, and thalassemia. Now we will learn about these hemoglobinopathies in details one by one. Okay, the first one, hemoglobin S. Okay, now this hemoglobin S will be formed when we have, see here, glutamate residue at position 6 in the beta chain. When it's replaced by valine, so this amino acid is replaced by valine amino acids, then it will result in the formation of hemoglobin S. Now, glutamate, the amino acid, is a polar one. It has a polar side chain. The valine, however, is a non-polar amino acid. Okay, so if such replacement is taking place, it will change the properties of the hemoglobin, because in a normal hemoglobin, it had a polar one in the outer surface, but now it has been replaced by a non-polar one. And as a result of these changes, the hemoglobin will now attract one another. That is, the other portion. Maybe one hemoglobin is abnormal, but there will be several hemoglobin that will be normal. So they will attract one another. And if such attraction takes place, it will lead in aggregation of hemoglobin molecules, because now they will be attached to one another, they will clump with one another. Okay, so they will bind with one another, clump with one another, which is not good. So they will look something like they are binding with one another, aggregated hemoglobin molecule. Usually in normal condition there is no aggregation of hemoglobin molecule. Now if hemoglobin aggregation takes place, this will result in the formation of a fibrous structure, and this will also result in changes in the shape of the RBCs. So if you remember, it's called HBS, right? So the RBCs will change into sickle shape. The S stands for sickle shape. So because of the aggregation, the RBCs structure are being distorted and they will look something like this. If you already know, a normal RBC will be biconcave, right? But in HBS, hemoglobinopathy or sickle cell diseases, we will have a sickle shaped RBCs. You can see the difference between a normal and a sickle cell RBC in this image over here. We know, or we have already studied, that when oxygen binds with hemoglobin, they are in a relaxed state, if you remember. Now, when we have such mutation which has resulted in substitutes of glutamine by valine, the relaxed state, okay, you'll see here, non-polar valine residues are not exposed on the surface in relaxed states. Okay, so therefore there will be no more aggregation. So what is the point here? If the hemoglobin is in deoxygenated form, it will promote aggregation. However, if they are in oxygenated state, that is their relaxed state, the non-polar portion of the valine is no more exposed outside. So if it's not anymore exposed, it will not attract other hemoglobin and will not cause aggregation. Okay, but deoxygenated, if the hemoglobin is in deoxygenated state, they will enhance the aggregation. Okay, so in oxygenated form, there will be no more aggregation in hemoglobin if the patient have sufficient amount of oxygenated hemoglobin. However, in deoxygenated form, we know that the hemoglobin will be in 10 state T. So the non-polar side of the valine will be exposed to outside. So they will attract hemoglobin and will promote aggregation. Now, hemoglobin will be present in deoxygenated form when oxygen tension is low. So there will be aggregation of hemoglobin S molecules and sickling of the RBCs if the patient does not have sufficient amount of oxygen. Okay. Now, if such happens, if there is aggregation, then there can be many problems here. One, as you can see in this image, they can block the blood flow. So all hemoglobin are causing the RBCs to turn into sickle shape, and therefore they will interfere with the normal blood flow as well. Okay, due to why? Due to abnormal hemoglobin. One more important thing is that the sickled RBCs can get destroyed early, premature destruction. Maybe they will not complete their 120 days lifespan. They will destroy early and may result in anemia. Okay, so these are the problems that can happen because of hemoglobinopathies such as HBS. The second hemoglobinopathy is our hemoglobin M, or also known as Boston hemoglobin. Now this one happens due to mutation where there is replacement of histone 58 in the alpha chain. So this amino acid is replaced by tyrosine. Now phenol group of tyrosine will bind with the iron, and as it binds with the iron, it converts the ferrous iron into ferric iron. We know the conversion of ferrous iron to ferric iron leads to something known as methemoglobin. And we also learned that methemoglobin cannot combine with oxygen. So patients who have such M hemoglobin, okay, or Boston hemoglobin, they will maybe suffer from low oxygen tension. Third and the last one is thalassemia. Now, thalassemia will result mainly due to decrease, lack of synthesis of either alpha or beta chain. It can also happen due to defective synthesis of alpha chain. If alpha chain is defective, we'll call it alpha thalassemia, and if beta chain is defective, we'll call them beta thalassemia. Now, if one type of chain, if there is a lack of synthesis of one type of chain, okay, this will cause overproduction of the other type. Let's say we have defective alpha chain, so it cannot be synthesized properly, so we will have excessive beta chain production. Now this will cause either a hemoglobin having only alpha chain or only beta chain. Okay, so one of them will be overproduced, other will have lack of production. Okay, next content is porphyria, another clinical aspect of the blood biochemistry lecture. We have already learned what is porphyria, porphyrin, and so on. Now porphyria, okay, not porphyrin, okay, porphyria is a group of disorder. Now mainly these disorders are due to mainly any abnormalities or defects in the synthetic pathway of porphyrin, basically. Now if anybody is suffering from porphyria, there will be large quantities of the porphyrins or their intermediates found in the urine. So mainly they will have a defect in the synthetic pathway, and because of that there will be excess amount of porphyrin found in the urine. So mostly it happens because of a defective enzyme. Okay, in the synthetic pathway, if we have a defective enzyme or absence of the enzyme, this can lead to porphyria. As a result of lack of enzyme or defect of enzyme, there will be intermediates formed in the synthetic pathway. Now, these urine in normal in color when fresh, but if you expose the urine of these patients of porphyria onto light, the urine will turn pink. mainly due to oxidation process. In the presence of light, there will be oxidations of porphyrogens. Patients who have porphyria also have skin sensitivity. When their skin is exposed to light, they also develop skin problems. The intermediates, as we have just seen, are the reason why these symptoms are appearing, because there is no enzyme, so there will be accumulation of intermediates. If the intermediates are in early phase of synthetic pathway, they will cause more problem or more severity compared to late intermediates, which are produced in the late stages of synthetic pathway. Because why? Because early intermediates are known to cause or damage to the nervous tissues, so they will have neuropsychiatric abnormalities. So for these porphyrias, the defective gene is basically found into a particular type of tissues. They will mainly cause expression in a particular tissues, and depending on their site of expression, we have two kinds of porphyrias: hepatic porphyrias and erythropoietic porphyrias. Hepatic porphyrias have again subtypes. We have acute intermittent porphyria, porphyria cutanea tarda, hereditary coproporphyria, variegate porphyria. Second one, erythropoietic have two subtypes: congenital erythropoietic porphyria and protoporphyria. Now let's check out each one of these. So under hepatic porphyria, the first one is acute intermittent porphyria. They are basically inherited in an autosomal dominant manner. The enzyme which have the defect is usually uroporphyrogen 1 synthetase, and the site of expression is liver cells. Second one, porphyria cutanea tarda. Autosomal dominant again. Enzyme that is involved is uroporphyrogen decarboxylase. Site of expression is liver cells. The third one, hereditary coproporphyria. Again autosomal dominant. Enzyme is coproporphyrogen oxidase. Expression is liver cells. Fourth one is variegate coproporphyria. Autosomal dominant. Enzyme is protoporphyrogen oxidase. Expression is liver cells. Now under the erythro, we have first one, congenital erythropoietic porphyria. See the difference? It's autosomal recessive. Enzyme involved is uroporphyrogen 3 co-synthetase. Site of expression is erythroid cells. Second one is protoporphyria, autosomal dominant. The enzyme involved is ferrochelatase, also known as heme oxygenase. Site of expression is liver cells. Now, in porphyria, the main signs and symptoms includes neurovisceral. If it's hepatic porphyria, it will mainly include, you see here, neurovisceral. In cases of erythropoietic porphyrias, it will mainly involve cutaneous, means skin. But there can be overlapping of symptoms in between these two types. Okay, so usually hepatic porphyrias patients will have acute attacks of abdominal pain, nausea, vomiting. Okay, in some cases they can have overactive sympathetic nervous system, so they may have tachycardia, very fast heartbeat, tremors, as well as hypertension. In some patients, they may also experience anxiety, insomnia, disorientation, depression, muscular weakness, and even seizure can happen. So mainly in cases of hepatic porphyria, the signs and symptoms include neurovisceral. Okay, so neurological sign and visceral sign mainly. In cases of erythropoietic, we have learned that cutaneous photosensitivity can be a feature. Okay, and as we have seen that there can be overlapping of symptoms, so you may find cutaneous photosensitivity also in hepatic porphyria, especially in hereditary coproporphyria, variegate porphyria, and porphyria cutanea tarda. You may have this skin manifestation as well. Now, the second kind, erythropoietic porphyria. In these cases, mainly they will have severe skin photosensitivity. So when their skin is exposed to light or sunlight, skin will be damaged. There will be vesicles formed on the skins which will erupt. Skin will become very fragile and pigmented. There will be areas in the skin which will have denuded. They will be more prone to infections. Some patients may also have bones and teeth involvement which will be pigmented, mainly due to deposition of porphyrin precursor. There can be destruction of blood cells due to binding of porphyrin to hemoglobin, okay, and liver damages can also take place. Even though liver is the site of expression for hepatic porphyria, but we have seen that these signs and symptoms can overlap in between them. So basically, the cutaneous manifestation will be formed when the patient's skin will be exposed to light. For neurovisceral symptoms, they can be precipitated by steroids, alcohol, and drugs. So if some people is taking alcohol, steroids, drugs, okay, and if they have the history of hepatic porphyria, they will develop the symptoms. The drugs that can initiate such actions will include barbiturates, carbamazepine, and so on. These drugs can cause the initiation of these symptoms. Okay, so these are the two types of porphyrias. So from this lecture, please do know the structures of the hemoglobin in detail. Okay, the subunits, the chains that we have. Okay, the important thing, know about the how the two status of the hemoglobin, the R status and the T status. So which one will bind to oxygen, how it changes, okay, and allows the oxygen to be released. Know about cooperative binding. Important thing is about derivatives of this hemoglobin, okay. Abnormal hemoglobin is the kind that we have learned today, hemoglobin S, hemoglobin M, and thalassemia. Know what is the problem in these abnormal hemoglobins, okay, and these hemoglobinopathies. And lastly, know about the porphyrias, that two kind that we have, hepatic porphyria and erythropoietic porphyrias. So that's all for today's lecture, and with this we have completed our blood biochemistry, and we'll continue the course in the next class. Thank you.